Hello everyone. Thank you for joining us for this exciting Q&A session titled Green Frontiers, Algae in Space Missions and Urban Sustainability. I am Magdaline Christina, research scholar here at JAIN deemed to be University School of Sciences. I am privileged to be speaking with Dr. Kumudini Satyan, a distinguished researcher, professor and program head of MSc Biotechnology here at the Department of Biotechnology and Genetics at JAIN deemed to be University School of Sciences. We welcome you, ma'am.
Thank you, Christina.
Her work bridges space biology, microbiology, and environmental sustainability. As we explore the fascinating role of algae and other extremophiles in the microgravity environment, we will also uncover how this research will help us to find climate-smart solutions on here, that is, on our Earth too. So let us quickly dive into knowing the impact of microgravity on algal growth and photosynthesis. Without any further delay, we would like to know, how does microgravity affect the growth and photosynthesis rate in algae or plants? And what could this tell us about its viability in life support systems for space missions?
Taking agriculture into space is not just an experiment. It is a promise that wherever humanity journeys, there will be seeds of life following it. So coming to your question on how growth and photosynthesis is affected by microgravity, yes, there are several ways in which microgravity affects. One of which is the disruption in gravitropism, where taking the cue from the gravity, the root grows downwards and the shoot grows upwards. But here when it grows downwards, because of the microgravity, there is no cue and the root growth will be completely disorganized, and that would affect the growth. The altered cell division and the rate of cell division would be affected. Other aspect that could be affected would be fluids that are present in the plant will not be settling down but will be in floating forms, such as the water droplets or films, which could affect carbon dioxide diffusion and leading to changes in the respiration. But overall, the photosynthesis is resilient. Although there might be some changes in the leaf orientation or the placement of the chloroplast, overall photosynthesis goes on. The photosynthetic mechanism functions. This has been observed in various ISS experiments too.
So could this also inform us about the climate-smart algae cultivation on Earth?
Yes, of course it does. One is the efficient use of resources and space. Then it is the fluid and gas management under controlled conditions, remote monitoring, and stress-resilient crop production. all these would definitely help in climate-smart algae cultivations.
Could you explain how microgravity can affect the gene expression and metabolic pathways in algae or cyanobacteria?
That's a very good question. Genes are expressed in various ways. Some are upregulated, some are downregulated. Some stress response genes, such as heat shock proteins, it may be reactive oxygen species, or chaperones which help with protein folding, are upregulated. Some photosynthetic related genes, such as the light harvesting gene or the photosynthetic chlorophyll synthesis genes, are downregulated. So the genes involved in cyclic electron transport can be upregulated. This is to maintain the ATP production under stress. Carbon metabolism. Carbon is the energy source. So carbon metabolism genes, they adjust. So the altered expression of enzymes that are necessary for glycolysis and Calvin cycle are upregulated. And transcription in genes supporting this alternative carbon storage is also upregulated. Photosynthesis and the carbon fixation pathways affects carbon dioxide diffusion. When it affects the carbon dioxide diffusion, there will be carbon dioxide depletion and oxygen buildup, which will help in increased respiration. In cyanobacteria, there are some specialized cells called the heterocysts. These heterocysts help in nitrogen fixation. That is why the cyanobacteria are powerful biofertilizers as they fix nitrogen. There are NIF genes or the nitrogen fixing genes that are present in these heterocysts. They differentially express under stress, disrupting the process of protein synthesis. One more possible gene expression change that could occur is a change in the metabolite profile. This affects the metabolic pathway. Here, osmolytes such as trehalose and proline increases. There could be an altered lipid synthesis, and there will be a change in pigment composition, especially carotene, which is a powerful antioxidant.
So does this help us understand the stress responses of these microorganisms during extreme conditions here on Earth?
Yes. This microgravity, what we are talking about, is similar to understanding cells under extreme stress conditions. These stress conditions overlaps with the terrestrial stress conditions such as deep sea conditions, hypoxic soil, drought, osmotic stress, and all this together has some real world applications too. It may be algal biorefineries, or in development of resilient crops, or in agriculture under controlled environment.
So shifting our focus to extremophiles, many space experiments have targeted or focused on tardigrades or algae. So what makes these microorganisms particularly suitable for space research?
Space is one of the environments which is most harsh. But these extremophiles are adapted to various stress environments on Earth. It may be desiccation, it may be radiation or oxidative stress. So all these stress conditions make them ideal organisms for stress studies. These organisms are also very simple. They have well characterized genome. It has simple well-characterized genome with known physiology. So this makes it very easy to carry out controlled, repeatable experiments. It also has different roles in oxygen production. It acts as a source of nutrition, and it helps in waste recycling. The ease of cultivation is very high. They can be easily cultivated in small volumes in compact bioreactors, which makes them ideal for experimentation in ISS.
Out of curiosity, we would like to know, are we studying similar organisms in our labs at JAIN?
Yes, we definitely are doing such experiments in JAIN, but under simulated stress conditions. We are working on bacteria and fungi under saline and drought conditions. In fact, we have a fungal isolate tolerating 1200 millimolar sodium chloride, which means it is similar to that of seawater. Algae under stress, as I told you, there will be a change in their metabolic pathway. Algae, they under stress conditions produced more lipids. These lipids that are produced can be trans-esterified into biodiesel. This is one of the important sources of alternate fuel, biodiesel. There are also studies on mechanical stress in plants and oxidative stress in blood cells that are being carried out. All these studies that are being carried out in JAIN have translational application.
In terms of climate resilience, how do the findings of space-based algal studies help us to develop sustainable technologies here on Earth, especially in terms of urban food systems and carbon capture?
Yes, algae can be grown in confined, controlled environment. Productivity can be maximized. Algal strains can be engineered to mitigate stress conditions and automate cultivation of algae. And in urban agriculture, it definitely helps in protein and micronutrient production, water efficiency, designing bioreactors, and also development of stress adaptive microorganisms.
Can this help us to combat from urban air pollution too?
Yes, in the form of biofilms, where carbon dioxide can be captured along with the volatile organic compounds in bioreactors. Bioreactors also help where these biofilms are used in bioreactors. It can help in capturing carbon dioxide as well as volatile organic compounds.
Finally, ma'am, what skills or areas of research should a student at JAIN focus on so that they can contribute towards similar interdisciplinary projects which involves space biology, microbiology, and environmental science?
Some of the important areas that they need to focus on is microbial physiology and genetics, where they generally understand the stress responses, model organisms and techniques such as qPCR or metabolite profiling. Plant and algal biotechnology concentrates on photosynthetic metabolism, carbon fixation pathways, and algal biotechnology. Then environmental biotechnology, where they study about bioremediation, biogeocycles, and microbial interactions. Molecular biology and omics studies includes cloning, CRISPR-based editing, transcriptomics, proteomics, and metabolomics, along with bioinformatics.
Thank you, ma'am, for sharing your valuable knowledge and expertise on developing a sustainable future.
Thank you, Christina.
I'm sure that the future researchers will implement the same ideologies for a better future on planet Earth and in space.